Welcome to the fascinating world of hemichordates. Imagine you are digging in the wet sand at the beach and you uncover a strange, soft-bodied creature that looks like a simple worm. However, this creature is hiding a massive evolutionary secret inside its squishy body. For a long time, scientists thought these animals were our direct ancestors because they share some very specific traits with us. Today, we are going to explore what makes them so special and how they serve as the evolutionary bridge between simple invertebrates and complex chordates like humans. Now that we have introduced our sandy friend, let us understand its place in history. Hemichordates are essentially the missing link on the tree of life. They sit right between simple invertebrates, like starfish, and complex chordates, which includes us humans. By studying them, we are literally looking back in time at the blueprints of our own biological history. They share specific traits with us, like structures for breathing and nerve cords, which is why early scientists mistakenly thought they were our direct ancestors. They are a crucial reference point for evolutionary biologists. Building on that evolutionary context, let us look at their formal biological definition. In biology, Hemichordata is a phylum of marine worm-like animals. They are bilaterally symmetrical, which means if you draw a line down their middle, the left and right sides are perfect mirror images of each other. They are also triploblastic, meaning their bodies develop from 3 primary embryonic cell layers. Finally, they are truly soilomate, which simply means they possess a true fluid-filled body cavity. These foundational traits are what classify them so uniquely in the animal kingdom. Understanding their basic classification leads us directly to their unique physical anatomy. A hemichordate's body is divided into 3 very distinct regions: a muscular proboscis at the front, a short collar in the middle, and a long trunk at the back. Along this body, they possess pharyngeal gill slits, which they use for both breathing and filter feeding. They also have a hollow nerve cord. However, the most critical distinction is that they lack a true notochord. Instead, they have a similar primitive structure called a stomacord, which is where their name actually comes from. To truly grasp this 3-part body plan we just discussed, let us look at a visual representation of a classic hemichordate, the acorn worm. Starting at the far left, you can see the muscular acorn-shaped proboscis, which the animal uses to burrow into the sand. Just behind it is the short, cylindrical collar. Extending to the right is the long trunk, which makes up the majority of the body. Notice the small openings along the upper part of the trunk, just behind the collar. These are the pharyngeal gill slits, used for filtering water and breathing. This simple but effective design has survived for millions of years. Having seen their anatomy, you might be wondering why we spend time studying a tiny, sand-dwelling worm. Their importance lies in their critical position on the evolutionary tree. Without hemichordates, we would have a massive gap in our understanding of how complex vertebrates evolved from simpler organisms. They show us how structures like the human spinal cord and respiratory system first began to develop millions of years ago. In the real world, studying their genetics and development helps scientists map out the fundamental rules of animal body planning, which even helps us understand human genetic diseases. Beyond their evolutionary significance, let us explore how these creatures actually survive day to day. One of their most important behaviors is filter feeding. Hemichordates pump seawater through those pharyngeal gill slits we saw earlier to extract tiny food particles. This process is not just good for the worm, it plays a vital role in cleaning the ocean sand and water. Because this is a physical process involving volumes and time, we can actually use mathematics to understand and measure how efficiently they interact with their environment. This brings us to our first mathematical model. To measure this feeding efficiency we just mentioned, marine biologists use the filtration clearance rate formula. Conceptually, this formula tells us how much water the hemichordate can process and clean in a given amount of time. The formula is C equals V divided by T. Here, C represents the clearance rate, usually measured in milliliters per hour. V represents the total volume of water filtered by the animal, measured in milliliters. Finally, T represents the time elapsed during the feeding process, measured in hours. If the volume V increases while the time T stays the same, the clearance rate C increases, meaning the animal is feeding much more efficiently. Now that we have our formula, let us look at a practical example to see how marine biologists study these creatures in the lab. Suppose a researcher observes a healthy acorn worm that successfully filters 150 milliliters of seawater over a continuous period of 3 hours. We are asked to find the clearance rate. We choose our formula, C equals V divided by T, because it directly relates volume and time to efficiency. We substitute 150 for the volume in the numerator and 3 for the time in the denominator. Dividing these gives us 50. This means every hour, this specific acorn worm filters and cleans 50 milliliters of seawater, showing its significant ecological impact. Just as we can measure their feeding, we can also measure their physical forms, a field known as morphometrics. Because different species of acorn worms look very similar to the naked eye, biologists need a reliable way to tell them apart. Instead of just guessing, they use specific mathematical ratios based on the animal's body parts. By comparing the lengths of different body regions, scientists can create a standardized, unitless number. This number can then be compared against a database of known species to confidently identify a mysterious worm found in the wild. To create that standardized number we just discussed, biologists use the body proportion index formula. Conceptually, this is a simple ratio that compares the front of the animal to its main body. The formula is I equals L sub P divided by L sub T. Here, I is the dimensionless body proportion index. The variable L sub P is the length of the proboscis, measured in centimeters. The variable L sub T is the length of the trunk, also measured in centimeters. Because we are dividing a length by a length, the units cancel out completely. If the proboscis grows larger relative to the trunk, this index value will increase. Let us apply this index formula to a real specimen. Imagine we find a worm where the proboscis length is measured at 2 centimeters and the trunk length is 10 centimeters. We need to calculate the body proportion index to help identify it. We use our ratio formula, I equals L sub P divided by L sub T. We place 2 in the numerator for the proboscis and 10 in the denominator for the trunk. The centimeter units cancel out. Dividing 2 by 10 gives us 0.2. This physical interpretation means the proboscis is exactly 1/5 the length of the trunk, a specific ratio that a scientist can look up in a biological field guide. To wrap up our journey, let us review the most important ideas we have covered. First, remember that hemichordates are vital evolutionary stepping stones, bridging the gap between simple invertebrates and complex chordates. Second, they are defined by a unique 3-part body plan consisting of a proboscis, collar, and trunk, along with pharyngeal gill slits. Finally, we saw how simple mathematical models, like clearance rates and body proportion indices, help us study their ecology and taxonomy. You have now mastered the fundamentals of Hemichordata, gaining profound insights into the history of life on Earth.